Okay, so this lecture is going to be on the pentose phosphate pathway or hexose monophosphate shunt. They both mean the same thing. The first thing we look at is what are the three pathways that cells can utilize glucose in. So you've already studied glycolysis, which is used to make ATP. You've already studied glycogenesis, storage of glycogen, which is an energy storage form when you're fasting. The pentose phosphate pathway is the third pathway, which can utilize glucose. And it's nothing to do with energy. So you may ask yourself that given that glucose is a nutrient, how is this pathway not using glucose for energy? So that's the first thing we'll look at. And then we'll go on to what are the two uses of the hexose monophosphate shunt or pentose phosphate pathway. I remember 3 years ago, this came in our SAQ, which is describe the two uses of the pentose phosphate pathway, or the two products of the pentose phosphate pathway. So I'll show you how to answer that. Then we'll discuss the pathway itself, the important steps. There's only two important steps you need to know. Then there's an important enzyme in the pentose phosphate pathway called transketolase, and its deficiency or its dysfunction causes Wernicke-Korsakoff disease or syndrome. You should understand what that is. The pentose phosphate pathway also generates NADPH, which has different functions in different cells. So that's the last thing we'll look at. So this is the first thing we should look at. Now, when glucose comes into the cell, it immediately gets phosphorylated by hexokinase in cells or glucokinase in the liver and forms glucose 6-phosphate. Now, this has three options. One is to go into glycolysis by conversion to fructose 6-phosphate to make ATP and then oxidative phosphorylation, etc. Then, alternatively, it can go into glycogen synthesis by going into through an enzyme called glucose 6-phosphate mutase, which converts G6P to G1P, which goes into glycogen synthesis. The last pathway glucose 6-phosphate can go into is called the pentose phosphate pathway. Now, as I said, glycolysis is used for generating ATP. Glycogenesis or glycogen synthesis is used for energy storage. The HMP shunt is used for neither. So this is the answer to the SAQ which we had, which is what does the HMP shunt produce? You should know two things. The first thing is it produces NADPH, which has different functions. The second thing it produces is something called ribose 5-phosphate. This is important because in MOL 2, you learn about synthesis of nucleotides, adenine, guanine, cytosine, thymine, uracil, etc. All of these are made from a precursor molecule, and that precursor is ribose 5-phosphate. All right. So if they ask you in the exam, in an MCQ or SAQ, what are the functions of the pentose phosphate pathway? You should say the function of the pentose phosphate pathway, firstly, is to produce NADPH. The second function is to produce ribose 5-phosphate, which is the precursor for nucleotide synthesis. Sure, I will send you the presentation. Okay. So these are the two functions you should be aware of. All right. Now let's go on to the biochemistry of the pentose phosphate pathway and the steps you need to know. So again, as I told you, glucose goes into glucose 6-phosphate. This can go into glycolysis. This is glycolysis. Alternatively, it could see an enzyme called glucose 6-phosphate dehydrogenase, or G6PD. Out of all the pentose phosphate pathway, all this pathway you see, this is the one thing you need to remember. All right, this step here. Glucose 6-phosphate goes to 6-phosphogluconate by the enzyme glucose 6-phosphate dehydrogenase. The dehydrogenase tells you what the enzyme does. It removes a hydrogen. It takes that hydrogen and puts it on to NADP to make NADPH. All right. 6-phosphogluconate then gets converted to ribulose 5-phosphate. You don't need to know this, but you should know this produces another NADPH. All right. So far, the pentose phosphate pathway, I produced 2 NADPH. Ribulose 5-phosphate, through a series of reactions, forms into ribose 5-phosphate, which I told you is the precursor for nucleotide synthesis. So this is the first function of the pentose phosphate pathway. This is the second function. All right. Now, ribose 5-phosphate can get made into other sugars, which can then go into glycolysis. You don't need to know the names of these sugars. All you need to know is that these reactions are catalyzed by an enzyme called transketolase. That's the only thing you need to know. All right. So your ribose 5-phosphate is the end product of the pentose phosphate pathway. It gets made into nucleotides. Alternatively, it can be made into other sugars, which can go into glycolysis. produce energy in the form of ATP. These reactions you don't need to know. The names of these molecules you don't need to know. The only thing you need to know is that these reactions are mediated by an enzyme called transketolase. Why is transketolase important? Because it requires thiamine. In your vitamins lecture, you probably studied that the active form of thiamine is called thiamine pyrophosphate. So that's what TPP is, thiamine pyrophosphate. This vitamin, this cofactor, is required by transketolase to function. And you should know that these pathways are essential for normal brain function. All right. So in a guy who's thiamine deficient, for example, this pathway won't work, and this guy will have abnormal brain function or encephalopathy. All right. So that's why this pathway is important. So the three things you need to know from the pentose phosphate pathway is firstly this step here, glucose 6-phosphate to 6-phosphogluconate by the enzyme G6PD. This produces NADPH. Eventually, through a series of reactions, you get ribose 5-phosphate, which can be made into nucleotides. Ribose 5-phosphate can be made into other sugars, which can go into glycolysis. You don't need to know the names of these sugars. I put an X here. The only thing you need to know is that these reactions are catalyzed by transketolase, which requires thiamine. This enzyme is essential for normal brain function. All right. Now let's look at transketolase in a bit more detail. You might recall in your other biochemistry lectures that there were a few other enzymes which required thiamine. Pyruvate dehydrogenase is an enzyme which converts pyruvate to acetyl-CoA. It requires thiamine. Alpha-ketoglutarate dehydrogenase is the rate-limiting step of the Krebs cycle. It requires thiamine. Branched chain ketoacid dehydrogenase is a part of the Krebs cycle. It requires thiamine. Does any of you know what happens if this enzyme is defective? You can comment or you can open your mic. What happens when this enzyme is defective? So yeah, so beriberi is what you get when you are thiamine deficient. Deficiency of this enzyme, like a genetic defect in this enzyme, causes a disease called maple syrup urine disease, which is valine, leucine, isoleucine. They end up in urine. All right. Now, okay, good. So the difference between transketolase and these three enzymes is that these three enzymes also require lipoic acid, coenzyme A, FADH2, and NADH to function. So these are TLCFN requiring enzymes. Transketolase is not a TLCFN requiring enzyme. It only requires thiamine, right? Now, there's a genetic disease where transketolase, it can't bind TPP or thiamine pyrophosphate normally. If it can't bind thiamine pyrophosphate normally, the enzyme doesn't function. And those pathways I showed you in the previous slide, they don't work. Hence, there's it affects and this affects the brain. All right. This disease is called Wernicke-Korsakoff disease. You don't need to know how it presents in patients. Just for your knowledge, Wernicke encephalopathy causes confusion. Patients become ataxic, they can't maintain their balance properly, and they get problems in their eye movements. Korsakoff syndrome is characterized by something called confabulation, which is where patients basically make up stories to fit holes in their history. All right. So you don't need to know that. All you need to know is that Wernicke-Korsakoff disease is a one genetic disease where transketolase has reduced affinity for thiamine pyrophosphate. All right. So now you might say, well, is this like beriberi? Thiamine deficiency doesn't cause Wernicke-Korsakoff disease. It causes beriberi, which is you do get dysfunction of this enzyme, but you also get dysfunction or impairment of this enzyme PDH, of this enzyme, and of this enzyme. So beriberi causes a lot of other stuff as well, in addition to Wernicke-Korsakoff. And so thiamine deficiency doesn't cause Wernicke-Korsakoff disease, it causes beriberi. And this causes Wernicke-Korsakoff syndrome, all right? This is not a genetic defect. The genetic defect is called Wernicke-Korsakoff disease. The pathology that you get secondary to thiamine deficiency is called beriberi. A part of that clinical syndrome is Wernicke-Korsakoff syndrome, all right? Does that make sense so far? Yes or no? Yep, okay. Okay, good. Now, so we've covered transketolase. All right, let me take you back to the objective slide. We've covered the three pathways utilizing glucose inside cells. I've told you the two uses of the HMP shunt. Can you remind me of those two uses? Number one was generation of something called NADPH. The second one was generation of ribose 5-phosphate, which is used to make nucleotides. Then I discussed the biochemistry of the HMP shunt with you. I told you that there's one step you should know, and that's glucose 6-phosphate to 6-phosphogluconate by the enzyme G6PD, and this produces NADPH. Then we covered the function of transketolase to mediate those series of reactions. You don't need to know the reactions. And you should know that this requires thiamine pyrophosphate. It's a genetic disease in transketolase, which reduces its affinity to bind to thiamine pyrophosphate. It causes Wernicke-Korsakoff disease. Wernicke-Korsakoff syndrome occurs secondary to thiamine deficiency as part of beriberi, all right? Now, this is the last objective. Now, let's look at the function of NADPH. NADPH, the function of NADPH depends on where you are in the body. In the liver cell, NADPH has a different function. In the immune cells, NADPH has a different function. In the red blood cell, NADPH has a different function. I'll first tell you its function in the liver. What was the enzyme that converts G6P again, if you don't mind? Yeah, sure, you should know this. Glucose 6-phosphate to 6-phosphogluconate by the enzyme G6PD, glucose 6-phosphate dehydrogenase. It takes a hydrogen from this glucose, puts it onto NADP to generate NADPH. Okay, you have to know this part. You have to know this stuff. All right. Now, I'll first tell you the function of NADPH in the liver, then in the neutrophil or in the immune cells, then in the red blood cell. In the liver, you see G6PDH, glucose 6-phosphate dehydrogenase, it produces NADPH. NADPH in the liver is used to make things, all right? It's used to make fatty acids, is used to make cholesterol, and it's used to make nucleotides. It donates this hydrogen to make nucleotides, right? The precursor of nucleotides is ribose 5-phosphate. As part of that metabolic pathway, or as part of that synthetic pathway, NADPH plays a role, all right? Now, okay, good. In the neutrophil or in immune cells, what is the function of immune cells like neutrophils or macrophages? They basically engulf bacteria into their cytoplasm, and then they generate stuff called reactive oxygen species. And these help in killing bacteria, right? You learn more of this in heme and in second year, but for now you should know that neutrophils and macrophages engulf bacteria or engulf microbes and generate reactive oxygen species, which allows them to kill these microbes, all right? Now we look at NADPH in this pathway, in this context, all right? So G6PD, the enzyme, generates NADPH. NADPH sees an enzyme called NADPH oxidase. This oxidase enzyme oxidizes NADPH. It takes the hydrogen and puts it on to oxygen here. And it takes this electron from NADPH, generating NADP, and it takes this electron and it puts it onto oxygen. Now this oxygen has a single electron. This oxygen now is called superoxide. And this superoxide is highly reactive. It's a reactive oxygen species. This superoxide runs into an enzyme called superoxide dismutase, or SOD. This produces hydrogen peroxide or bleach. So the bleach you see in your house, that's hydrogen peroxide. This hydrogen peroxide is used to kill bacteria. All right. Now, it should make sense to you now that people who are G6PD deficient, who are deficient in the enzyme, they are immunodeficient, and so they're at an increased risk of developing infections. The second way you could get the same an increased risk of infections is that if you're defective in this enzyme, NADPH oxidase, and you should learn this in your free radicals lecture, your last lecture, I think, the free radicals and reactive oxygen species. Can you repeat the NADPH oxidase part? Okay, sure. But first, let me finish. NADPH oxidase, its deficiency causes a disease called CGD, or chronic granulomatous disease. If NADPH oxidase doesn't work in CGD, you can't generate hydrogen peroxide, hence you can't kill bacteria. And so these patients who suffer from CGD, or chronic granulomatous disease, they suffer from recurring infections. G6PD deficiency could give you something similar, all right? Where does the H of the protein go to? Can you repeat? Okay, I'll repeat the function of NADPH in the neutrophil, right? So neutrophils and macrophages are immune cells which engulf bacteria. They generate reactive oxygen species, which allows them to kill the bacteria, all right? G6PD, the enzyme we discussed, generates NADPH. It has a hydrogen here. NADPH runs into an enzyme called NADPH oxidase. This oxidase enzyme takes this hydrogen, there's this electron, and puts it onto oxygen. This produces a molecule called superoxide, which is highly, highly reactive. Superoxide runs into an enzyme called superoxide dismutase, which converts superoxide to hydrogen peroxide or bleach, and bleach kills bacteria. So there's two ways you could get immunodeficiency. One is you could be G6PD deficient. This will decrease the amount of NADPH you have in your body. This will decrease this reaction here. Hence, you can't get hydrogen peroxide. Hence, you can't kill bacteria. The second way you can get immunodeficiency is that if you're deficient or defective in this enzyme. Chronic granulomatous disease, or CGD, is a genetic defect in NADPH oxidase. This enzyme doesn't work, hence you're not able to produce superoxide. If you're not able to produce superoxide, you're not able to produce bleach, hence you're not able to kill bacteria. And so in G6PD deficiency and in chronic granulomatous disease, you get immunodeficiency with increased risk of infections. Does that make sense? The people who were asking for it to be done again? All right, excellent. Now the question comes, how do you distinguish between G6PD deficiency, this is G6PD, not GDPD, G6PD deficiency, and chronic granulomatous disease? What other things does G6PD deficiency cause which distinguishes it from CGD? Now, the two things you should know are Heinz bodies and hemolytic anemia, right? So now this is the pathway you should know, and this is the major pathway of the whole presentation, right? So I'm going to explain it to you on a whiteboard first. Now, imagine you have a red blood cell here, and red blood cells are basically bags of hemoglobin, right? They just have a lot of hemoglobin in their cytoplasm. They don't even have nuclei, all of it's hemoglobin. They go into the lungs, they pick up oxygen, oxygen saturation goes up to 100. They go out into the arteries to your fingertips, where they release the oxygen. Then they go back up into the lungs, pick up the oxygen, oxygen binds to hemoglobin. Then they go out into the periphery and drop off the oxygen, all right? So they're just bags of oxygen, RBCs, all right? Hemoglobin is like the only important thing inside RBCs. The other important thing inside RBCs is the membrane. If the membrane doesn't work, what will happen? Water will come inside the cell, and the cell will swell up and it will burst. So the red blood cell needs ways to protect hemoglobin, and it needs ways to protect the membrane of the red blood cell. All right, so that's the big picture you have to know, right? So the red blood cell is, hemoglobin is an integral, essential for the function of the red blood cell. The red blood cell membrane is obviously essential to the function of the red blood cell. So the red blood cell needs to protect these two things. But what does it need to protect these two things from? It needs to protect hemoglobin and its membrane from reactive oxygen species. So let me give you an example. You all should know from basic chemistry that O2 plus 2 electrons goes to form O2 minus, right? And this is stable. But what if oxygen only gains a single electron? This will form superoxide, the thing we looked at previously, right? This is superoxide. It has an oxygen. It's an oxygen with only one electron, which only gained one electron. This means this oxygen is highly reactive. It needs another electron. It wants to get another electron. I also told you that this can get converted into bleach, right, by an enzyme called superoxide dismutase. And hydrogen peroxide is also very, very reactive. Now, how does hydrogen peroxide become stable? It gets two hydrogens and it becomes water, right? So hydrogen peroxide is very, very reactive, and it will do anything to get these two hydrogens, all right? So this reaction is very, very important to you, and I think you should probably know this from basic chemistry, this reaction of hydrogen peroxide, these two hydrogens. Now, let's suppose you have hydrogen peroxide inside the RBC. Hydrogen peroxide will need these two hydrogens, will need these two protons here. But how will it get those two protons? Where will it get them from? I'll show you a reaction. Now, obviously, you know hemoglobin is a protein. Proteins are made up of many amino acids, and one of those amino acids is cysteine. Cysteine, as you know, as you should know, has a sulfur group as its R group. So I'm drawing a hemoglobin here with a cysteine sticking out, the group of cysteine sticking out. I'll draw another hemoglobin here with another cysteine sticking out, right? So what hydrogen peroxide will do is take these two hydrogens here and form a disulfide bridge between these two hemoglobins, all right? So that's what's gonna happen. Obviously, you know that disulfide bridges determine what is one of the determinants of the tertiary structure of a protein. So by doing this, hydrogen peroxide alters the tertiary structure of the protein, alters the tertiary structure of hemoglobin, and makes that hemoglobin non-functional and makes the red blood cell useless. So you will see, for example, if hydrogen peroxide is allowed to act on hemoglobin, it will keep linking these hemoglobin molecules together. You see that? Does this make sense to everyone? This is very, very important for you to know. Does it make sense so far? Yeah? All right. So obviously now your red blood cell needs a way to combat this, to protect hemoglobin. So it has a decoy molecule. That decoy molecule is called glutathione, all right? This is the molecule called glutathione. What is glutathione? Glutathione is a tripeptide. It's 3 amino acids. The middle amino acid is cysteine. So I'll draw a G here, and I'll draw an SH here, and you know what's gonna happen, right? And I'll draw another G here, another SH here. So glutathione will get oxidized and give these two hydrogens to hydrogen peroxide. It will form GSSG, which is oxidized glutathione. This is reduced glutathione, all right? This reaction is mediated by an enzyme called glutathione peroxidase, all right? Now, so this way the red blood cell can save hemoglobin from hydrogen peroxide by sort of sacrificing glutathione. Glutathione is giving the protons to hydrogen peroxide, and it's saving hemoglobin. But now, obviously, you're gonna have more hydrogen peroxide. So you need to find a way to reduce oxidized glutathione back into GSH, right? Back into 2-GSH. So this was 2 glutathiones. I need to find a way to make this oxidized form back into the reduced form. So there's an enzyme in the red blood cell called glutathione reductase. Glutathione reductase basically adds 2 hydrogens back to glutathione to reform reduced glutathione. The next question you should ask is, where do you get these 2 hydrogens from? You get these 2 hydrogens from NADPH, all right? So as I told you, the pentose phosphate pathway generates 2 NADPH. In the red blood cell, those 2 NADPH molecules donate their hydrogen to oxidize glutathione to reform reduced glutathione. This reduced glutathione can now continue giving hydrogens to hydrogen peroxide. And this way, glutathione, or the red blood cell, is protected, hemoglobin is protected from hydrogen peroxide. Does that make sense to everyone? Because this is the major concept of this lecture. So it makes sense you've understood the lecture. Can you repeat the last part? Okay. Now, glutathione is basically a decoy that gives off these 2 hydrogens to hydrogen peroxide. It becomes oxidized in the process, right? It forms a disulfide bridge. I need to find a way to add 2 hydrogens to this oxidized form to regenerate this reduced form. So now I can continue dealing with the hydrogen peroxide. How do I do this? I need to find a way to add 2 hydrogens. The 2 NADPH that you generate from the pentose phosphate pathway are used by glutathione reductase. Glutathione reductase takes the hydrogen from NADPH, puts it on to oxidize glutathione, and remakes reduced glutathione. this can now continue dealing with hydrogen peroxide and protect hemoglobin. Now, I hope that's clear. Now you should ask yourself, in a patient who is G6PD deficient, what will happen to reduced glutathione? So let me ask you, what will happen to reduced glutathione in a patient who is G6PD deficient? Will it increase or decrease or stay the same? What will happen to the reduced form of glutathione? It will decrease, exactly. Good, excellent. If the reduced form of glutathione decreases, now this reaction, now hydrogen peroxide can go and attack hemoglobin, and it can link hemoglobin molecules together by disulfide bridges. And also hydrogen peroxide then damages the lipids and proteins. It does the same thing to the proteins in the cell membrane, in the RBC membrane. Hence, the RBC membrane gets leaky. It becomes leaky, water comes into the cell swells up and it bursts. So you have something called hemolytic anemia. Heme to do with the RBC, lytic, RBC is burst, all right? And anemia, you have low RBCs. Now, the special feature of hemolytic anemia caused by G6PD deficiency is that if you see the red blood cell under a microscope, if you take a blood sample from a patient who's G6PD deficient, and you look at their blood under a microscope, and you look at their RBCs, you will see these collections of hemoglobin. So these collections of hemoglobin linked together, you will actually see them inside the RBC, and they are called Heinz bodies. All right, they are called Heinz bodies, like the ketchup. All right. Now, so that's the concept you should know from this entire lecture. That's the most important thing. In a patient who is G6PD deficient, hydrogen peroxide will oxidize the hemoglobin, link it together, forming Heinz bodies. Hydrogen peroxide will also damage the membrane, and this will cause lysis of RBC as water comes in. Does that make sense to everyone? So let me ask you now. So we previously looked at the fact that G6PD deficient individuals and people with CGD both have immunodeficiency. But how do you distinguish the two? In G6PD, you will have Heinz bodies and hemolytic anemia. I hope that makes sense now. Now, you might ask yourself now that where am I getting the reactive oxygen species from? Why is hydrogen peroxide in the red blood cell? Now, why does the red blood cell have hydrogen peroxide in the first place? Now, what you should understand is that the red blood cell goes into the lungs where oxygen tension is very, very high. This forces the oxygen onto the hemoglobin, specifically the iron of the hemoglobin, iron which is Fe2+ or ferrous iron, right? Now, when the red blood cells go into the periphery, into your capillaries and in your tissues, they will obviously drop off the oxygen. But some of that oxygen, about 0.5 to 3% of the oxygen, will take an electron from Fe2+. This will form Fe3+. Hemoglobin with an Fe3+ iron is called methemoglobin, METHB. And the oxygen, because it's now gained one electron, is called superoxide, which is what we discussed. Superoxide runs into an enzyme in the red blood cell. This is the red blood cell, right? Erythrocyte. Superoxide runs into an enzyme called superoxide dismutase, which forms hydrogen peroxide. Also, some oxygen is also spontaneously converted to hydrogen peroxide in the red blood cell. So these are where you get the hydrogen peroxide from. All right. Now, look here. Glutathione is reduced, or GSH, which is what I wrote down, right? GSH runs into an enzyme called glutathione peroxidase. It gives off those two hydrogens to form water, sort of neutralizing hydrogen peroxide. And in the process, it forms oxidized glutathione, or GSSG. Now you need to find a way to recycle glutathione in order for you to keep dealing with the hydrogen peroxide, because hydrogen peroxide is continuously being generated, right? So you need a continuous way of recycling this glutathione back into the reduced form. So there's an enzyme called reduced glutathione reductase, which takes the hydrogen from NADPH, puts it on to oxidized glutathione, and reforms reduced glutathione. This allows the red blood cell to keep dealing with the hydrogen peroxide. In a patient who is G6PD deficient, there's an arrow here, right? In a patient who's G6PD deficient, NADPH will go down. The reduced form of glutathione will go down, as you said. Hydrogen peroxide, hence, will accumulate. Then this hydrogen peroxide will start attacking the hemoglobin. This will link the hemoglobin together through disulfide bridges. This will also, hydrogen peroxide will also damage the membrane, causing hemolytic anemia. If you look at the RBCs of these patients, you will see these collections of hemoglobin inside the red blood cell. We call these collections Heinz bodies, all right? That's the concept here. Now, so G6PD deficiency is a very, very common disease. You will see it in probably in your clinical practice in the future, inshallah. It's an X-linked recessive disorder. Now, people who are G6PD deficient, they don't have, it's not like they have no G6PD. It's just the activity of G6PD is reduced compared to the normal person. The activity of G6PD is reduced. So they have a low amount of NADPH. This low amount of NADPH, and this will cause a low amount of reduced glutathione. But even this low amount of reduced glutathione is enough to deal with normal amounts of hydrogen peroxide. So individuals with G6PD deficiency are actually normal at birth, and they're mostly asymptomatic. They live normal lives because even though their reduced glutathione stores are reduced, there's still enough to deal with the normal amount of hydrogen peroxide, which is seen in the red blood cell, which is 0.5 to 3%. However, there are certain conditions which can increase hydrogen peroxide production. And this will overwhelm the glutathione, causing hemolysis, hemolytic anemia with Heinz bodies. There are certain infections can increase. Infection is a stress response, right? And in a stress response, free radical generation and superoxide generation is increased. And so these infections will increase the amount of hydrogen peroxide. Certain drugs that you learn in second year will also increase the amount of hydrogen peroxide. The one thing you have to remember is fava beans, right? Fava beans, people consume them in the Mediterranean region quite a bit, and they actually increase hydrogen peroxide production. So the classical scenario that you probably will get in your exam is a normal patient who's living a normal life. He consumes a meal, and all of a sudden he develops hemolytic anemia. And when they see his blood under a microscope, they see Heinz bodies. Then they'll ask you a question about that. That's the classical scenario of a G6PD deficient patient. It's an X-linked recessive disorder with reduced activity of G6PD, which causes reduction in the amount of NADPH you're producing, and hence the stores of glutathione that you have are reduced. This is enough to deal with normal amounts of hydrogen peroxide, but things which increase hydrogen peroxide production, like fava beans and infections and certain drugs, they will cause hemolysis and Heinz bodies by overwhelming the supply of glutathione. All right. This is just, I've listed to you here the presentation of hemolytic anemia, just in case they don't give you the word hemolytic anemia in your exam. You should be able to recognize that this patient is suffering from hemolytic anemia. Anemia manifests clinically with pallor. Patients are pale, shortness of breath, because anemia is low RBCs, right? So you can't get enough oxygen to the tissues. So your ventilation, your breathing rate increases to increase the oxygenation of the blood. Fatigue, because the oxygen supply is reduced to the muscle in anemia because you don't have enough RBCs. This causes fatigue. Jaundice, because when the hydrogen peroxide is attacking the membrane of the red blood cell is bursting open, it's releasing hemoglobin into the bloodstream. Hemoglobin, a part of hemoglobin is converted to something called bilirubin. Bilirubin causes jaundice. Splenomegaly, most of the hemolysis which goes on occurs in the spleen, and so the spleen gets big. All right. So I've mentioned this just in case they don't give you the word hemolytic anemia. You should know that patients with hemolytic anemia get shortness of breath, palpitations, fatigue. They also develop jaundice. They can develop enlarged spleen, splenomegaly, enlarged liver, hepatomegaly. All right. Now, this is an interesting question for all of you. What is the other disease which causes hemolytic anemia that you guys have studied in biochemistry? There's one more disease you guys have studied in biochemistry, which causes hemolytic anemia. So G6PD is the most common one. There's another one. Does anyone know? Okay. There's a disease called pyruvate. There's a disease called pyruvate kinase deficiency. Excellent. Yeah. Pyruvate kinase deficiency. I'm just telling you this because it will allow you to sort of integrate your knowledge a bit better. So the last step of glycolysis is phosphoenol pyruvate, right? And this is mediated by an enzyme called pyruvate kinase, and this step generates ATP, right? Now, this step will actually generate 2 ATP because, as you should know, that when fructose 1,6-bisphosphate gets broken down, and then you get 2 molecules of glyceraldehyde 3-phosphate. So all the steps below that step in glycolysis are multiplied by 2. So if there's generating 1 ATP, you'll actually have two of these reactions going on, which means you'll generate 2 ATP. This allows you, this means that glycolysis actually generates 4 ATP. 2 ATP are used by glycolysis, and so the net gain of ATP is +2. So you're actually gaining 2 ATP in glycolysis. All right. However, when this pathway is defective, what will happen? You will lose those 2 ATP. Hence, what will happen is you'll only generate 2 ATP here, and your net gain of ATP will be 0. So the red blood cell can't carry out oxidative phosphorylation because it doesn't have mitochondria. So the only way the red blood cell gets energy is through glycolysis. And in pyruvate kinase deficiency, because this step doesn't work, you're not getting ATP, and so you're not getting any energy. So now the question is, what are the consequences of low ATP on the red blood cell? Why does the red blood cell need ATP? Does anyone know? Why does the red blood cell need ATP? All right, so the red blood cell needs ATP because in the membrane of the red blood cell, there's something called sodium-potassium pump. All right, so, and these are found in the membranes of all cells, and these require ATP. All right, this requires ATP. Now, let's say the red blood cell is not generating ATP. It's generating 0 ATP because of pyruvate kinase deficiency. What will happen to these pumps? They'll stop working. So what will happen to sodium inside the cell? Sodium will start accumulating inside the cell. That means osmolality inside the cell will increase. That will drag water into the cell. The red blood cell will swell up and it will burst, giving you hemolytic anemia. So pyruvate kinase deficiency and G6PD deficiency, they both give you hemolytic anemia, but the mechanism is completely different. In G6PD deficiency is because of hydrogen peroxide. In pyruvate kinase deficiency is because of lack of ATP. Does that make sense? All right, excellent. I told you that because they might confuse you in an exam with a hemolytic anemia patient, and you will say, okay, this guy has G6PD, but he might have pyruvate kinase deficiency. So let me cover differences in the clinical presentation of G6PD deficiency and pyruvate kinase deficiency. I told you that G6PD patients usually are completely normal, and they only develop hemolysis whenever they get an increased stress, such as fava beans or such as infection. Pyruvate kinase deficiency, or PKD, it occurs in newborns. You'll see a newborn child, which in the question, if they give you pyruvate kinase deficiency, they'll say this newborn child developed pallor, shortness of breath, fatigue, symptoms of anemia. He developed jaundice and splenomegaly. Then you should think about pyruvate kinase deficiency. If it's a normal man who just eats a meal and develops hemolysis, think about G6PD deficiency. Also, let's say a guy has hemolytic anemia and you take a blood sample and you look at the blood under a microscope and you don't see Heinz bodies. If they say this patient's peripheral blood has no Heinz bodies, think of pyruvate kinase deficiency. Does this make sense? It's slightly complex, but I hope you guys are following along. Does this make sense to everyone?
Can I ask a question?
Sure.
You said that in pyruvate kinase deficiency, there will be no Heinz bodies.
Yeah, there won't be Heinz bodies.
Isn't it one of the clinical presentations though?
In G6PD deficiency, you have Heinz bodies. In
Okay.
pyruvate kinase deficiency, you don't have Heinz bodies. In both, you have hemolytic anemia.
Okay.
In pyruvate kinase deficiency, you have hemolytic anemia because the ATP is deficient, and so the sodium potassium pump isn't working. So water is coming into the cell, so the cell bursts, right? In pyruvate kinase deficiency. In G6PD deficiency, that is because hydrogen peroxide is attacking the membrane and it's causing the membrane to become leaky. And this causes the water to come into the cell and the cell bursts. So this causes hemolytic anemia. But additionally, in G6PD deficiency, the hydrogen peroxide is also attacking the hemoglobin and it's linking those hemoglobins together, right? And this forms, right?
Yeah.
In pyruvate kinase deficiency, this doesn't happen. So in pyruvate kinase deficiency, you don't see Heinz bodies.
Okay,
all right?
Thank you.
And so these are some Heinz bodies for you guys to look at. This is the peripheral blood smear, where when they look at the person's blood under a microscope, and you see these collections of hemoglobin. If you see these collections of hemoglobin, these are Heinz bodies. This is because of G6PD deficiency. All right. So in summary, this lecture, I started off telling you that glucose can be utilized in 3 ways: glycolysis to generate ATP, glycogenesis to store energy, or the HMP shunt, which has nothing to do with energy. The function of the HMP shunt is to produce NADPH and ribose 5-phosphate, which is the precursor for nucleotide synthesis. And important, then I covered the biochemistry of the pentose phosphate pathway with you guys. I told you that there is only one reaction that you have to know, which is glucose 6-phosphate to 6-phosphogluconate catalyzed by an enzyme called glucose 6-phosphate dehydrogenase, or G6PD, and this generates NADPH. Then I told you that ribose 5-phosphate, the end product of the pentose phosphate pathway, can be made into other sugars, which can go into glycolysis. You don't need to know the names of the sugars. All you need to know is that those reactions are mediated by an enzyme called transketolase. This requires thiamine. It doesn't require PLCFN. It doesn't require lipoic acid coenzyme A, FADH2, NADH. It only requires thiamine. And this enzyme is essential for normal brain function. A genetic disease in patients, a genetic disease where transketolase can't bind thiamine pyrophosphate properly, this causes Wernicke-Korsakoff disease. Alcoholics, on the other hand, alcohol inhibits thiamine absorption. So alcoholics can develop beriberi. A part of beriberi will be Wernicke-Korsakoff syndrome, not the disease, the syndrome only. All right. The syndrome means genetic defect. How do you treat this? You give thiamine. All right. Now, I'll get to this later on. When I finish the summary, I'll get back to this point with an interesting clinical scenario for you guys. Then once we were done with transketolase, we covered NADPH. I told you the functions of NADPH depend on where you are in the body. In the liver, NADPH is used for synthesis of fat and cholesterol. In immune cells like neutrophils or macrophages, NADPH is the hydrogen from NADPH is used by an enzyme called NADPH oxidase to produce superoxide, which then produces hydrogen peroxide, which is bleach, and this kills bacteria. Then I told you that patients who are G6PD deficient can become immunodeficient, and patients with chronic granulomatous disease, where the NADPH oxidase deficient, people with G6PD deficiency can become immunodeficient, and people with chronic granulomatous disease have lack of function of NADPH oxidase, so they can also become immunodeficient. How do you distinguish between the two? G6PD would also cause hemolytic anemia with Heinz bodies. All right. In the red blood cell, NADPH is used to recycle reduced glutathione to allow neutralization of hydrogen peroxide, which protects hemoglobin and the red blood cell membrane from oxidative damage. All right. Individuals with G6PD deficiency may develop hemolytic anemia with Heinz bodies on exposure to stressors like fava beans. You need to remember fava beans. And you need to distinguish hemolytic anemia from G6PD deficiency against hemolytic anemia secondary to pyruvate kinase deficiency. Pyruvate kinase deficiency also causes hemolytic anemia, but it causes hemolytic anemia because of loss of ATP. So you can't maintain those active transport mechanisms on the cell membrane. All right. Now, let me get back to you with a question to do with thiamine, to do with your vitamins lecture. So now you guys know that thiamine causes beriberi, and you know that alcohol inhibits thiamine absorption. So alcoholics are predisposed to developing thiamine deficiency. Okay. Alcoholics also, they don't take care of themselves properly. And so a lot of times in the West, an alcoholic will present to the ER with hypoglycemia. So let me give you a clinical scenario. An alcoholic presents to your clinic with hypoglycemia. You give the guy glucose to correct his hypoglycemia, but the guy dies. What did he die from? What do you guys think? Beriberi? Okay. Okay. So you're kind of there. Now, you know that acetyl-CoA pyruvate, it gets converted to an enzyme that gets converted to acetyl-CoA, which is the first part of the Krebs cycle, by an enzyme called PDH, right? PDH. And this requires TLCFN, but concentrate on the T, concentrate on thiamine. The other pathway that pyruvate can go into is, what other pathway can pyruvate go into?
Anaerobic respiration.
Excellent. Yeah. So it can go into lactic acid production. Okay. Lactic acid production. Excellent. Now, let's suppose that alcoholic that you just gave glucose to also is thiamine deficient. If he's thiamine deficient, this pathway won't work. And so all of the glucose that you gave the guy will go into lactic acid production. And so he'll develop lactic acidosis, and he could die from lactic acidosis. Excellent. Yeah, exactly. So when you're in a clinic in the West, what they'll teach you is that if an alcoholic comes to your clinic, you should always give the guy thiamine with the glucose. You shouldn't give the guy glucose straight away. Otherwise, you could kill him because of lactic acidosis. Does that make sense to you? Yeah. All right. So we're done with the lecture. If you guys want, I've put some questions down for you guys. So if you want to do them, you can. So this is a 14-year-old boy who was brought to the physician by his mother because of a 12-hour history of abdominal pain and dark urine. 3 days ago, he had a cold. Examination shows jaundice and splenomegaly, enlargement of the spleen, and pallor. A peripheral blood smear, which is when they look at the blood under a microscope, shows inclusions within erythrocytes like this one, right? The underlying cause of this patient's condition is most likely to also affect which of the following processes. He has G6PD deficiency, but you have to be careful while answering this question, right? So which of the following pathways which will also be defective in this patient? What was the other pathway we discussed in G6PD deficiency? Other than the red blood cells, we discussed something in immune cells, right? So de novo synthesis of glutathione, you can synthesize the glutathione, but he just can't recycle the glutathione. So there is actually not a problem with synthesis of glutathione. Excellent. Yeah, it's B. All right. Does everyone understand this? So he had a problem generating superoxide. So all of this scenario was about hemolytic anemia. But what they asked you is that what other process could be affected? Generation of superoxide could be affected. Could be affected in this patient because NADPH oxidase requires NADPH from the pentose phosphate pathway to generate superoxide. Now, conversion of phosphoenol pyruvate to pyruvate, what is that? That is pyruvate kinase deficiency. Anchoring of proteins to the cell surface, you will learn this in heme. This is called hereditary spherocytosis. All right, but you can see the Heinz bodies. You can see that the patient just had an infection, which was what we discussed, right? Infections causes increased hydrogen peroxide production, which causes hemolytic anemia. So this boy was completely normal until he developed the infection, which increased stress and increased hydrogen peroxide production, which caused the hemolytic anemia. All right. Now this contrasts the previous case now with this case. This is a male newborn baby born to a 27-year-old woman. Within the first 24 hours of birth, he develops hemolytic anemia with jaundice, hepatomegaly, splenomegaly, and cyanosis. Laboratory studies, you forget about this, no Heinz bodies seen in peripheral blood smear. Which of the following is likely involved in the pathogenesis of this patient's conditions? Excellent. It would be A, because this is pyruvate kinase deficiency. All right. Decreased in reduced form of glutathione, that's G6PD deficiency. Point mutation in beta globin gene, that's sickle cell anemia. Osmotically fragile red blood cells, that's hereditary spherocytosis. Forget about that. You learn about that in heme. So no Heinz bodies. So pyruvate kinase deficiency. Yep. Excellent. So both cause hemolytic anemia. Pyruvate kinase doesn't cause Heinz bodies. So whenever they describe a patient with hemolytic anemia, you should always think about pyruvate kinase deficiency, G6PD. Then you should look at whether there's Heinz bodies. If there are Heinz bodies, the guy has G6PD. If not, the guy has pyruvate kinase deficiency. Also, G6PD patients, also you can get a hint of the answer by the scenario. Look at the previous scenario here. This was a 14-year-old boy who was completely normal until he developed the infection, because G6PD patients are completely normal until they develop a stress. So that stress can be fava beans, infections, drugs, or in contrast, pyruvate kinase deficiency patients are always symptomatic. Even at birth, they develop hemolytic anemia. All right, so if you see hemolytic anemia in a newborn, it's likely pyruvate kinase deficiency. If you see hemolytic anemia in an adult, all of a sudden, it's likely G6PD deficiency. All right, so we're done with the lecture. Any questions? Clear? Would it be enough if we just studied from your slides? Yeah, I've covered the important things. So what I would suggest is, because the pentose phosphate pathway is so complex, look at the video a couple of times. We didn't take that much time. You can speed up the video just to get an idea of sort of how the pathway works, what are the different uses of NADPH, how does glutathione function, that whole pathway of reduced versus oxidized glutathione. It'll take you just some time to get used to that. All right. I hope that was clear. Okay, thank you.